This video is going to get you through the protein synthesis notes. This chapter, I've got some other videos that discuss DNA and how those get used, so if going through this you still had some questions about it, you might try some of those other videos. You can also find some really good videos out there at Khan Academy and Cash Course that show you the process of DNA replication and transcription and translation, and so please watch those because you have to understand this in a lot of detail, so make sure you spend the extra time to learn this that you need to. To start out, we're going to talk about, again, important people. Gregor Mendel is considered to be the father of genetics. He was an Austrian monk. He was trained in mathematics, and so the numbers as he was breeding peas specifically is what helped him understand how traits get passed down from one generation to the next. The way your book describes his work is that he discovered that traits are inherited as physical units. We did not know about DNA at this time. We didn't know about alleles at that time. We knew a little, we knew about cells, and we knew that cells had a nucleus, but we didn't know whether DNA or proteins were the genetic material. We didn't even really know about chromosomes at that time. And so he's one of the people who first started to say it's a physical thing that's getting passed down from one generation to the next. We now know that those physical things are genes present in the DNA, but he was one of the people who was a stepping stone that got us to that knowledge. Next, we've got what did George Beadle and Edward Tatum figure out? Usually we're just going to refer to them as Beadle and Tatum. They work together. They were working with a mold called Neurospora crassa. What they would do is they would grow the mold in different media. Now, if you provide the mold with every single nutrient that they need, it can grow and flourish. What they would do is they would hit the mold and then mutate them so that they were unable to use some of the nutrients that were present in the medium, and then that would prevent them from growing. They knew that in order to use nutrients, you needed enzymes to help you work with those nutrients. Remember previous stuff on microbial metabolism? They knew that. And so they came up with what it was known at the time as the one gene, one enzyme hypothesis. And what that essentially meant was genes code for enzymes. We now know that genes don't just code for enzymes, but again, they were getting us towards that step on what do genes actually do? Those traits that you're passing down from one generation to the next, how do they make you who you are? And they believe that they just coded for enzymes based on their experiments with that mold. This is just kind of a refresher, high school stuff. You should be able to recognize what DNA looks like at this point. It's double helix. It's got a sugar phosphate backbone. It's got nucleotides that kind of form ladders where you get rungs to a ladder, and there's hydrogen bonds that attach adenine to thymine and cytosine to guanine. So this is a DNA molecule. Okay. Getting down to the anatomy of DNA. So DNA is a nucleic acid. Nucleotides are the monomers that make nucleic acids. This is one specific nucleotide. There's 3 components to it, and ATP is technically a modified nucleotide, so we got a lot of the same groups. P.S. Spelling error, I did not make this picture. So the nucleotides are going to consist of 1 phosphate, not 3, like ATP. That's the modification that ATP has. We've got a sugar. The sugar is going to vary depending on whether the nucleotide is going to be for RNA or DNA. If it's for RNA, it's ribose. If it's for DNA, it's deoxyribose. And then we've got 5 different nitrogenous bases that we're going to be talking about. That's what makes it an A, a T, a C, a G, or a U. That's adenine, thymine, cytosine, guanine, or uracil. It's called a nitrogenous base because there's lots of nitrogen that's present in the nitrogenous bases. The rest of this is mostly carbon and hydrogen, but again, we do have that phosphorus over here in the phosphate group. This just shows you some of the other nitrogenous bases that can be present. Some of them are modified, but uracil is one of them, guanine is one of them. The other ones are all modified, but there's that. A nucleobase. What I tell you there is that most books call that just the nitrogenous base. So the nucleobase is the nitrogen-containing part of the nucleotide. A set of 3 nucleotides codes for an amino acid. When you're talking about a set of 3 nucleotides on mRNA, that's going to be called a codon. We'll talk about that a little bit later on. A string of amino acids makes a protein eventually. Essentially what we're going to have is multiple steps here. If you put 2 amino acids together, you get a dipeptide. If you put 3 or more amino acids together, you get a polypeptide. When the polypeptide takes on the shape that it's supposed to have by doing the primary, secondary, tertiary, and quaternary structure, that's when you get a protein. So a protein is essentially a folded polypeptide. What are some of the functions of proteins? They do everything in your body that is supposed to happen in your body. We've already talked about enzymes. The majority of your enzymes are going to be proteins. They can be structural components. Like they make up parts of the cytoskeleton. They also, in multicellular organisms, can make up stuff like ligaments and tendons and hair and things along those lines. They can help with different processes that are supposed to happen, or transport of materials, like those channels and carriers that are present in the cell membrane. Those are going to be proteins. They can help with cell-to-cell recognition. They can do a whole slew of things, but essentially, if there's a function that's supposed to happen in your body, it's a protein that does it. So one of the questions that I usually like to ask here is, so how does DNA make you you? Well, your DNA is what provides you with the recipes for making all of your proteins. And again, the proteins are what do everything inside of your body. So that's how your DNA makes you who you are. It provides you with a recipe book for how to make every ingredient that your cells need to be able to make. The genome is essentially your whole entire cookbook. It's every single recipe that's present inside of the cell. In terms of a bacterial cell, this is an E. coli that has been lysed and all this stringy stuff out here. That's their chromosome. That's their genome. Any plasmids that are present in that cell would also be part of their genome. A gene is just a segment. It's one recipe to cook one particular polypeptide or ingredient. The way your book describes that is it's a section of DNA that encodes a specific product. And again, usually that's going to be a protein, but it could also be a specific type of RNA. There's other things genes can code for, but we're usually going to talk about it in reference to that protein. Genomics is the study of the nucleotide sequence that's present within an organism, and so this would be like popping open the cell and not just looking at, hey, look, there's a chromosome, but it's, let's look at the sequence of it. What is the sequence of letters that exists within that recipe, so we can find out what does a normal cell look like versus what does a mutated cell look like. Briefly explain DNA replication, and by briefly I did mean briefly. We're going to go into it in more detail later on, but for right now it's just DNA duplication. You take one molecule of DNA and then you copy it so that you're going to end up with two molecules of DNA. DNA replication has to happen before any cell multiplies, so this happens during the process of binary fission for prokaryotic cells, and it happens prior to mitosis and meiosis in eukaryotic cells. Briefly explain gene expression. So gene expression involves two steps, transcription and translation. Gene expression is also often called protein synthesis. What we do is we read the recipes present in the DNA and we make copies of them into messenger RNA. That messenger RNA leaves the nucleus for a eukaryotic cell, or in a prokaryotic cell it just goes to find a ribosome, and then that ribosome reads the recipe in the form of that messenger RNA, and then it uses that recipe to cook the dish, which is the protein. So we copy DNA into RNA during transcription, and then we read RNA to make a protein during translation. So transcription is just that process that copies one or a few genes during transcription to make a copy of messenger RNA. During transcription, we do not copy the entire genome. We just copy the genes that we actually need to make at that time. Translation reads the RNA to actually make the protein. That happens at the ribosome. There's some other molecules that are going to help us out. Most notably, tRNA or transfer RNA is going to help with the process of translation. So essentially, transcription is copy the recipe. Translation is cook the recipe. Said another way, transcription copies DNA into RNA. Translation reads RNA to make a protein. After that, you have described the structure of DNA, including all of the following terms. This is where you need to spend a lot of time looking at a molecule of DNA and really understanding how that molecule gets made. It is two nucleotides, or nucleic acids, pardon me, that are going to be put together. One polynucleic acid, that's going to be a sugar, a phosphate, and a nitrogenous base, and they're going to get stacked together. What you end up with is what's known as a sugar-phosphate backbone. Again, if you kind of think of this like it's a ladder, the sides of the ladder are the sugar-phosphate backbones. The rungs that you would step on, that's where the nitrogenous bases are. The sides are all held together with covalent bonds. So your sugar, covalent bonds. The phosphate is covalently bonded to the sugar. So this is a very strong molecule because covalent bonds are the strongest type of bond that could be present. The nitrogenous bases, on the other hand, are held together with hydrogen bonds. Remember, that's the weakest kind of bond that we make in our cells. That means we can split apart these two strands really easily, and that's going to be very important for DNA replication and transcription, because when we do that, we have to read one strand at a time, so we have to be able to pull them apart. So know where we've got covalent bonds and where we have hydrogen bonds, because that's relevant for how DNA ends up getting used. Now, when you start talking about chemistry, anytime you've got a fork in a road, essentially that's a carbon. So there's a carbon here, a carbon here, a carbon here, a carbon there, a carbon there. The carbons get numbered. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. The reason why that's relevant is that tells you why you have the 3' and the 5' ends of a DNA molecule. The 3' end is coming off of the third carbon within that ribose or deoxyribose. Since we're talking about DNA, this would be a deoxyribose sugar. The alcohol group, which is this OH, comes off that third carbon, so that's the 3' end of the molecule. The phosphate, on the other hand, comes off the 5th carbon in the sugar, and so that's why the phosphate side is the 5' side. So it's all about which carbon are we at in the sugar molecule. Now, one strand of DNA is going to go, in this case, from 5' to 3'. The other one runs in the opposite direction. Notice it's upside down, so it's 3' to 5'. That is what's known as anti-parallel. So the two strands of DNA run in opposite directions to each other. Notice how the complementary base pairing works between one strand and the other strand. So A base pairs with T. There's 2 hydrogen bonds to that. That means A's and T's aren't as strongly attached to each other as guanines and cytosines or G's and C's. There's 3 hydrogen bonds that hold those together. That's relevant because the origin of replication, where replication starts in a standard bacterial chromosome, it's rich in A to T. That means it's easier to break the molecule apart at that place because there's fewer hydrogen bonds that are holding those molecules together. Let's see, anything else that I need to say there? Usually I have students try to build some molecules of DNA in class for this. You can do that with some pipe cleaners and some beads just to kind of make sure you understand it. But make sure you understand all of the structure of DNA. Again, videos online can really help you understand that. Next, describe the structure of RNA. So RNA is also a nucleic acid. It's also made out of nucleotides, but generally speaking, your RNAs are going to be single-stranded, not double-stranded. So we're not going to do the complementary base pairing in a standard RNA molecule. We'll do it to make an RNA molecule, but not as the molecule is completed, it won't stick around, essentially. So this is a molecule of messenger RNA. It still has a sugar phosphate backbone. That's the zigzaggy line that you have off down here. It still has nitrogen-containing bases sticking off the side. One of the big differences between DNA and RNA is there are no thymines in RNA. So wherever there would have been a thymine, a uracil replaces that. So we have uracil, cytosine, adenine, and guanine are the nitrogenous bases that make up RNA. There's 3 different kinds of RNA that we're going to talk about. There's actually way more than that, but we're only going to talk about these 3. There's messenger RNA. That's a copy of a recipe that gets read by a ribosome, which is this whole structure. There's tRNA, which are these green, oddly shaped molecules that are in the ribosome in this picture. tRNAs are called transfer RNAs because what they do is they transfer amino acids to the ribosome. So amino acids, again, are the recipes for making proteins. I like to call tRNA the sous chef because it brings the ingredients to the chef being the ribosome. The third type of RNA is rRNA. That's ribosomal RNA. That's actually part of the ribosome. Ribosomes are made out of RNA and protein, and the RNA that makes them up is called rRNA. Anything else I want you to know there? I think I pretty much covered everything. Transcript is just a copy of a gene from DNA that hasn't been modified just yet. Eventually the transcript is going to become messenger RNA. I said all the rest of that jazz. Describe the function of the following. I really hit those earlier, but once again, messenger RNA, that encodes a protein. It's a recipe for a particular protein. tRNA is what I usually call the sous chef. That's transfer RNA. It transfers amino acids to the ribosome. And then rRNA is ribosomal RNA. That's actually part of the ribosome. Okay. Completely understand how DNA replication takes place, why it occurs, and what enzymes are necessary to carry it out. And then I give you some definitions just to make sure you kind of understand everything. I'm going to show you this video. Once again, this is from a non-majors textbook, but it can show you the moving parts that are relevant for DNA replication, and then we'll talk about it afterwards.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, so let's see. I was hoping I had some pictures of some of that stuff, but I'll just use the video for some of the pictures. So to start you out, replication fork, that's the place where DNA helicase splits the two strands of DNA apart from each other. And so notice there's two replication forks anytime helicase splits it, and that's because we're going to kind of split in the middle of the chromosome and then open in both directions. We do bidirectional DNA replication. Semi-conservative replication. What that means is that whenever we make molecules of DNA, we don't destroy the old strand. Instead, we use that old strand as a template to build a new strand. The new molecule of DNA is going to be half old DNA and half new, so we conserve the old stuff. That's why it's called semi-conservative replication. The origin of replication, that's the place where helicase first starts to split to create the replication fork. Bacterial cells just have one origin of replication within their single circular chromosome. Our chromosomes that are linear, they actually have multiple origins of replication. DNA polymerase. Now here's another place where majors need to know a little bit more detail than the non-majors. There's two different DNA polymerases that are going to be relevant for DNA replication. DNA polymerase 3 is the one that is going to be adding nucleotides after the primer, which this red section is the primer. So then DNA polymerase 3 adds nucleotides following the rules of complementary base pairing to the template strand. So this kind of brownie orange one, that's DNA polymerase 3. Non-majors, you don't have to know DNA polymerase 3 versus the next one I'm going to talk about. Let's see. Gotta find the right one. Alright. This yellow one that comes in, it removes the primer and then it adds nucleotides that are DNA. Now, here's the thing about the primers. They're RNA nucleotides, not DNA nucleotides. That's because DNA polymerase 3 can't just start replicating. It has to have an end to start from. And so the primer serves as that end for it to continue the process of replication. So what needs to happen is the primer has to be removed, and then we have to add nucleotides that are DNA nucleotides where those RNA ones had been. So a different DNA polymerase called DNA polymerase 1 comes in to remove the primers and then add DNA nucleotides after that. So majors, there's 2 DNA polymerases. 3 builds their original strand, and then 1 comes in to replace the primer. And then for non-majors, just DNA polymerase builds new strands. The leading strand is the strand that can be built continuously from start to finish. In the picture or the movie that I showed you, this top strand was the leading strand. So DNA helicase keeps unzipping in this direction. And so this strand is being built in this direction, so we can just keep building following the DNA helicase with no problem. The bottom strand, on the other hand, since we're unzipping in this direction, we're actually building in this direction because it's anti-parallel, and so we have to keep starting over again. So we'll build from here, and then we get to another primer. That will have backed up, and so now we can start again. So we build it in little fragments that are called Okazaki fragments. So Okazaki fragments are the fragments that the lagging strand is built in. Complementary base pairing, what you need to remember is A to T, C to G. So that's as we're reading, and there was actually a picture of that kind of in here. Let me see if I can find that. There we go. There's how the complementary base pairing works. So if the template strand has a G, DNA polymerase 3 is going to add a C to the new daughter strand that's being built. If there's a T, we add an A, and so on. This is, again, one of those things where watching lots of videos can really help you understand what's happening in the process of DNA replication. Alright, so if the picture represents DNA replication, what's the name of the green enzyme? That green enzyme looks like it's building a new strand from it, so that would be DNA polymerase. Which strand in the picture represents the leading strand? So remember, the leading strand gets built continuously from start to finish, so this top one would be the leading strand. This bottom one down here, that's the lagging strand, and you can tell because it's made in little fragments. So there's an Okazaki fragment, there's an Okazaki fragment. Those are eventually going to be glued together with DNA ligase. Adenine can only base pair with what when replicating with DNA? So adenine base pairs with thymine. D is the right answer for that. This is a picture from, it's not actually from your book now that I'm looking at it, it's from a journal. It shows you the origin of replication. It's called OREC in E. coli. So this is the place that's rich in A to T because there's fewer hydrogen bonds there. So DNA helicase first comes in here and starts to unzip the 2 strands. So we generate those replication forks, and then we just build in both directions from that origin. Alright, next up, completely understand how genes are expressed. So this again is protein synthesis. Transcription and translation is what you're going to have to remember on here. Notice that I once again recommend that you watch some videos, and so here's a video to help you understand from a non-majors textbook.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
We won't look at protein processing, that's just the process that folds it, and it happens in the rough ER, generally speaking, and prokaryotes don't have that, so we're just going to kind of skip that section. So coming back to make sure you understand the definitions that are present, first one for you guys is RNA polymerase. That's the enzyme that carries out transcription, so it reads the DNA and it makes the mRNA. It does make other types of RNA too, but usually we're going to talk about it making mRNA. As it does that, it is important for you to understand it's only reading one strand of DNA, the one that contains the gene. It's not reading both strands simultaneously, so just doing one. The promoter is the place on the gene where the RNA polymerase first binds so that it can start that process of transcription. So right about there, we'll back up a little bit. Right about there would be the promoter, since that's where the RNA polymerase is first binding to get that process started. The promoter has a specific sequence that helps it recognize, and there's usually stuff called growth factors there, or transcription factors, that help it recognize that this is where it's supposed to get started so it can start that process of reading. Think of it sort of like it's the capital letter at the beginning of a sentence. That's how it knows start here and read through here, because we don't want to start in the middle of a gene, then we don't make the whole entire protein. Terminator, that's the end, that's like the period at the end of the sentence that tells the RNA polymerase it's done so it can leave. Complementary base pairing works very similarly to how it did in DNA replication. The difference is if there's an A present on the DNA, it would like to base pair with T, but there's no T's in RNA, so instead it's going to base pair with uracil or a U. So T can still go to A because we still have adenines in RNA, but A can't go to T anymore, it has to go to a uracil. G's and C's still work exactly the same, so that's the difference in complementary base pairing. After that, during translation, so tRNA and the ribosome do a really good job of bringing the right amino acid and then growing the polypeptide, which is the purple thing that's working there. But in order for you to be able to do that, you have to use something called the genetic code. This is the genetic code that's usually put into textbooks. It's the box format. In order for you to read the genetic code, first thing you have to know is you have to be reading messenger RNA. Messenger RNA, a series of 3 nucleotides, is called a codon, and so you translate a codon using the genetic code. So let's say you had the codon, I'll make it easy, CCC. The first letter in the codon, C, is on this side, so C. Second letter is across the top, C. Third letter is across the right side, C. So you find the place where all of those intersect. So C, 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 codes for pro, which is proline. And so proline would be the amino acid that the tRNA brought to the ribosome to start making that protein or to get that process going. Now there is also a different representation that they never put in textbooks. I think it's because it's easier to use and they don't want to make your life any simpler. But in order to use the circle genetic code, you still have to read mRNA, you still have to have your codon. You start in the center of the circle and you work your way out. So C, 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 codes for proline. It gets you to the exact same place. In my opinion, it just gets you there that much sooner. You do not have to memorize the genetic code, but you do have to be able to use it. I usually put the box form on the test because that's the version that's in your textbook, and I want to give you what you have easier access to. So make sure you're able to read the box form of the genetic code. Let's see. Maybe I don't have things for that, so I'll kind of come back to this. A codon is a set of 3 nucleobases present on mRNA. So like CCC could be a codon. AUG as a codon, if you use the box to read that, AUG, it codes for the amino acid methionine or MET, but what's important about it is it's also the start codon. So it's the first amino acid that always gets brought that tells the ribosome, here's where you're supposed to start reading. The reason why that's relevant is if you remember back to the video, there's a cap that's made out of a modified guanine that gets added to the beginning of mRNA. That's not part of the recipe. We don't want to read that. So what happens is the ribosome and the tRNA, it skips the first part of the mRNA until it finds an AUG that codes for methionine, and then it knows this is where the recipe starts and I should continue to read from that point on. So AUG is the start codon that codes for methionine. Next, UAA, UAG, and UGA, those are all stop codons. That's the period at the end of the recipe. Again, that's what tells the ribosome that it's done cooking that dish. After that, I said be able to read the genetic code. Again, be able to read the box. Reading frames are what you guys have next. I'm not sure if I'm going to have a really good picture to go along with reading frames. I'll kind of come back to this. Okay, so this was the transcript that got made earlier. It has the modified guanine cap and then it has a poly-A tail on this side. Well, remember that we don't read the beginning of the messenger RNA. That's sort of just something for the ribosome to grab onto. That AUG is what sets the reading frame for a particular strand of mRNA. I'm going to doodle on this screen to kind of show you what I'm talking about. Sure, I'll do it in red. Okay, so we've got modified guanine cap. Then we're going to have just some stuff. That continues on with that mRNA sequence. Well, what sets the reading frame, remember, we don't read until we get to the start codon. So what would happen is the ribosome comes down here and doesn't, ooh, weird, don't know how I did that. Anyway, doesn't read this jazz off down here until it gets to AUG. I got to figure out how to undo what I did a second ago. Once it gets to AUG, that's what sets the reading frame. And from there, we start reading this 3 letters at a time. So first we read AUG, that would code for methionine. Then we skip to the next set of 3 letters, CUA. We would figure out what that codes for. So just for fun, we'll skip over to this. What does CUA code for? You can use either one to try to read it. Hopefully you got there. I'm going to show you on this one. So CUA codes for leucine. So we would come back. So this one added a MET. This one added a leucine. The ribosome would bond those together. Then we'd skip to the next set of 3, AGU, and we'd read that. AGU codes for serine, and if you wanted to do it over here, AGU still codes for serine. So we'd come back and we'd add the serine. The ribosome would bond those together. We'd keep reading until we hit a stop codon. If something happens that shifts the reading frame, it makes the recipe entire gibberish that doesn't turn into a functional protein in most cases. And so what sets the reading frame is that start codon. And then from there, we just read it 3 letters at a time. Okay, Ribosomes, that's what acts as a chef. It reads the recipe and then it cooks the protein, and so the blue in this picture would be the ribosome. tRNA is what I called earlier the sous chef. It brings the ingredients, the amino acids, to the ribosome so the ribosome can do the cooking. The tRNA has an anticodon. tRNAs are specific. Each one can only carry one kind of amino acid. And so if AUG is the codon, the tRNA would have the complement to that. And so this tRNA right there would have the anticodon UAC, and then it would attach to this AUG. And that's how we specifically bring the MET to get us going at the very beginning. So yeah, the anticodon is present on the tRNA. It attaches to the codon. That's how we know which ingredient to bring in there. After that, you have what are some of the differences between prokaryotic and eukaryotic gene expression. So in the video, they showed you a cap and tail being added. Prokaryotes don't do that. So prokaryotes don't add the cap and tail because they don't need the help to get out of the nucleus like a eukaryotic cell is going to. So we don't add the cap and tail to prokaryotes, just eukaryotes. One of the other things that we've discovered at this point is that in typical eukaryotic genes, we have exons, which in this picture are yellow. Those are the actual coding regions, but because we've been infected by a lot of viruses, viruses insert their genome into our chromosomes, and so we end up with little bits of viral stuff or little bits of gibberish in between our coding regions. Well, the gibberish is called an intron. What we do in eukaryotic gene expression is we splice out the introns and then glue the exons together to make our actual functional mRNA. That's called mRNA processing or splicing when that happens, and only eukaryotes do that. Prokaryotes don't. Next, signal transduction. There's lots of chemicals and things that happen outside of a cell that are trying to tell that cell something like nutrient availability or, hey, there's too much waste that's accumulating out here. That signal needs to be relayed from outside the cell to inside the cell, and that's what signal transduction is. So it's transmission of information from the outside of the cell to the inside. Usually a receptor is going to play a role in that. Incidentally, receptors, they're typically proteins. Quorum sensing is a really interesting thing that cells are able to do. What they can do is essentially tell a cell how many neighbors it has in a particular place. If you've never had to participate in committee meetings, one, you're lucky, but two, a quorum means that you have a minimum number of people so you can go ahead and have that meeting. If you don't have a quorum, the meeting just gets called off. Well, prokaryotes can sense a quorum and then change their behavior if a quorum is actually present. And if the quorum's not there, they don't do that behavior change sort of a thing. One of the things that can happen, and I don't remember if I have the hyperlink on here. I don't on this one, but hopefully I'll have it in just a little bit. There's a bacterial species that's called Vibrio fischeri. It's a species that lives out in the ocean. If they're just a few cells floating around, they just float around in the water and they live their lives, they process their nutrients, life is all good. But if there ends up being a bunch of Vibrio fischeri in one place at one time, they can sense, hey, we have a quorum now, let's make life interesting and throw a party, and they turn on genes that allow them to actually glow or bioluminesce. And so you might have seen pictures where you can see a bioluminescent beach where the waves that come up are actually glowing. That's because of things like Vibrio fischeri. They start to glow when there's a bunch of those species present in one place, and they only do that at high cell density, so that's quorum sensing. Next, you have explained how an operon works. An operon is essentially an on-off switch that allows a cell to know, I need to read this recipe and cook it, or, hey, we don't have enough cells present, we should not be reading that recipe. There's certain parts to an operon. So first off, we have the promoter. Remember, the promoter is the place where RNA polymerase can bind to a gene and then carry out transcription and copy that gene. The operator is the part that actually acts like a switch. If there is a repressor bound to the operator, then RNA polymerase can't attach to the promoter and then zip down here because essentially there's a gate that's up and it can't get past that gate. And so when the repressor's bound, these genes are turned off or they're silenced because we don't need to do that right now. One of the first operons that we discovered is the lac operon that's present in E. coli. That's what this picture is trying to represent. So E. coli lives in your gut. If you are not a dairy eater, that means you don't give your body a lot of lactose, which is the sugar present in dairy products. Well, why do they need to make enzymes to break down lactose if you're not providing them with any lactose? A repressor binds to this and they stop making those enzymes to break it down. But if you did just have breakfast maybe and there was milk in your cereal, you've provided the E. coli in your gut with lactose. The repressor will detach from the operator. They'll go ahead and transcribe these genes and then start that process of helping you digest the lactose that was in that milk that you just drank with your breakfast. So the operon is essentially just a way for them to turn on or turn off different genes. Again, a lot of this is based on different proteins that are present in the cell. Now for this, I have a link to a video that shows you not just how operons work, but also how that quorum sensing from just a second ago works. And so here's this video.
So what you're seeing first, this is just DNA in the bacterial cell, and we've color-coded the genes. So that's the Lux operon. There's 5 genes in that operon, and somewhere else on the DNA is encoded the gene for that master protein LuxR in purple. So the first thing that's going to happen is that RNA polymerase, that's RNA polymerase, the green protein, is going to come to the DNA encoding LuxR. It gets transcribed into a messenger RNA, that's the molecule you'll see now, and then ribosomes come onto that messenger RNA and they translate the message into protein, right? So now the pink protein, that's LuxR, the master transcription factor getting made in the cell. When that protein gets made, which is only at high cell density, it's going to go and tell all these genes to get turned on so the bacteria can switch into quorum sensing mode. We're showing you it getting on to the Lux operon. Its job is to tell RNA polymerase, come here, come here, come here. This is a gene that we want to make the protein now. So it's sitting on the Lux operon. Here comes RNA polymerase. And then it's going to transcribe this long operon. There's 5 genes on this messenger RNA, so they're in colors for you to see this. And then the ribosomes get on at each gene, and they translate the message into proteins. We're just showing you 2 important proteins that are called Lux-A and Lux-B. Those are 2 proteins that fit like locks and keys, and when they get made at high cell density and come together, they give off photons of light. So they lock in together and light gets produced, which is what you saw in the demonstration. But of course, these bacteria are machines. It's not just one, transcript getting made. There are thousands of copies of LuxR on thousands, turning on thousands of messages to get made so that the cell can make thousands of copies of luciferase so that there's enough of those Lux-A and B proteins coming together that the entire cell actually makes perceivable light. And so that's what we're showing you there. And then if we zoom out of the cell, remember this is high cell density, so the outside world of the cell, those autoinducers are there, the bacteria know they're at high cell density, they want to switch into quorum sensing mode, enough luciferase gets made that the cells turn on light and you can see it.
Alright, so hopefully that helps you understand how quorum sensing works. And now I need to shrink that down and come back to Operon. Okay, after that you guys just have some model organisms, starting with... Oh, I clearly did not turn something off that I should have. Hold on. Darn TED Talks always self-playing in the background. Alright, now I gotta go back to where I was. Alright, E. coli is your model organism that you have. Now, E. coli is one that I mentioned just a second ago. It is a gram-negative species. While it is part of your normal flora, there are certain strains that can cause disease, or if it gets into a place where it's not supposed to be, it can cause disease. So while it's supposed to be in your large intestine, it should not be in your urethra. So one of the more common infections it can cause is a UTI, which is a urinary tract infection. It can also cause diarrhea if you can get some of those strains that you're not supposed to necessarily have in your body. What shape does it have? You should be able to tell me that at this point. It's a rod shape. What's the word for that? It's a bacillus. What is the toxin produced by the enterohemorrhagic strain of this species? It's called the Shiga toxin, S-H-I-G-A. Shiga toxin breaks down cells that line the blood vessels, and enterohemorrhagic means you start bleeding into your intestines. It gives you bloody diarrhea, which can be fatal. Only some strains of E. coli have that toxin though, and so it's very important for you to understand, again, most E. coli is okay as long as you're keeping it in your intestine, but those toxin producing strains are not good. Your book mentions a specific operon that has been studied in this organism. Well, this is the same one that I gave you a picture of a little bit ago. It's the lac operon, LAC. What does the, same slide. What does the above operon control? The ability to transport and digest lactose. Under what conditions would the repressor be present for that promoter? When conditions are low lactose. So when they don't need to digest lactose, that's when the repressor would be attached. Under what conditions would the repressor become non-functional? If there is lactose available, then they do want to digest lactose, so the repressor would go away so that RNA polymerase could zip down the genes and then go ahead and transcribe them. After that, I have that little activity to help you carry out DNA replication, transcription, translation. I would recommend that you go ahead and practice doing that. If you want to, you can show me your work next time we meet in lab, or if we're done with lab, you can send me your stuff in email just to make sure you understand it if you want to. I do have some additional little worksheets that you can do. If you want some additional help with protein synthesis, just email me and I'll give you some stuff that can help you out with this. But don't forget to take the quiz that's posted in school.